That we have two distinguished speakers in this
अरे ये
session,
पेन ड्राइव जा रहा
one
है
before lunch and one after lunch. So the
किसको
first
कॉपी
talk
करना है
is by Dr. Subeer
कहाँ
Majumdar,
पर भी इसको लगा के
who
डायरेक्ट
has
चलाओ
been in Hyderabad for a
मेरे
long
कॉपी
time.
पेन ड्राइव से
He
डायरेक्ट
headed the National Institute of Animal Biotechnology.
कॉपी
and,
मत करो डायरेक्टली
I
चलाओ
think. Subeer, you were founder director or no?
नो सर
after retired.
प्रोफेसर या प्रोफेसर रेडन
But he transformed that campus which we have seen.
3 होगा देखो
And right now he has moved from Hyderabad as Director
कॉपी
General
मत करना कॉपी
Gujarat Biotechnology University, Gandhinagar. And he has been known for his animal biotechnological experimental techniques.
Can you keep
I
that
think he
here?
is one of
That's
the
my only
authorities
request.
in India.
keep it here.
And then another major contribution from him is he has developed
because
uh,
pointer जब
chip
नहीं चल रहा है
to
the
identify
pointer doesn't
Indian
work, I can go up and down
breeds of
With
cows.
up and-
They have a big
like
collection.
it doesn't work
And I think this chip is being developed and quite likely it will come into use. So he's a fellow of all the 3 science academies and he has been a recipient of Tata Innovation Award and JC Bose Fellowship. So it's my pleasure to request Subeer to give his talk.
so good आ गया presentation coming okay I can go do from here also because it's so good afternoon so I will try to work from here because this one is very low and these are very bright so it is better to use the computer and use the arrow here so that you can understand well. So first of all, I thank you, the sectional committee as well as INSA for giving this opportunity to me to speak. And as I said that I am going to talk about biotechnology, human and animals, and how animals are important for helping human and how human should help animals. So I worked at National Institute of Immunology, National Institute of Animal Biotechnology, and at present as Gujarat Biotechnology University. And these are all my colleagues who has helped me in showing, in helping whatever I am going to show. So we all know, and Chandrimati has shown in the morning, that how the whole world is really concerned with the major infectious disease outbreaks. So although if you try to check the outbreaks in Southeast Asia, it appears India has less. It is not so because surveillance is less, and that is why we do not see major output, but definitely there are a lot of cases. So there are a lot of diseases which are coming from animals to human beings, and I have named the animal groups. They can be wild, they can be pets, or they can be domestic animals. And from hospital, from a patient, it can go to a doctor, or in a place, it can be from animal to human, human to human, human to animal, and animal to animal. So it's complicated. It is not that easy. And somebody asked question to Chandrimati from where it came. God knows. So there are a lot of zoonotic diseases, whether it is influenza, salmonellosis, West Nile virus, plague, emerging coronavirus, rabies, brucellosis, Lyme's disease, and now CCHF is coming from Pakistan, from our left side of the border. Leptospirosis, we know Mumbai and Kerala very often gets, and bovine tuberculosis is there. So from human to animal and animal to human, this tuberculosis also jumps. Along with that, another thing we are facing is antimicrobial resistance. So most important is the disease detection and detecting it correctly and quickly. It should not be false detection. So some of the biotechnological things which our institute at NIAB took as a lead, which I kind of told them to do this kind of work, has really helped. So we hired a person with biosensor, or it's an animal biotechnology institute, but the person didn't have anything. Then we hired a person from aptamer specialist. And we tried to see that unless the detection is good in the animal, so we need the technologist, not necessarily the animal biologist, and to address these issues. So I will show some of them. So for example, when you do the diagnosis of brucella, so many human patients are admitted in hospital. So doctors usually confirm diagnosis of brucellosis by testing blood or bone marrow for brucella bacteria, or by testing the blood and antibodies to the bacteria. To help detect complication of brucellosis, your doctor may also order additional tests like X-rays, etc., to see several changes in bone joints. So ideally, the IgG goes and stays up to 6 months, but the IgM can stay up to 2 years. So what if you want to epidemiological study, you have to catch what is there in, do you have a traces of antibody in the body or not. The problem with the animals is government gives you vaccination to be done to the animals, and animals are vaccinated mostly by the surface LPS related vaccines, and when you go to check whether there is a trace of brucellosis in, or an outbreak of brucellosis somewhere, you find positivity in animals which are vaccinated as well as animals which are infected. So you need to differentiate infected versus actually vaccinated. So our institute started doing those kind of work, and we got a protein array from USA and actually took those proteins which are inside the brucella and which comes out as a secretory product. And if you have, we generated kits for that, so that if you have antibody against them, you are actively infected with brucella. But if the animals surrounding have antibodies for the LPS, it can be vaccinated or it can be really infected. So this is a very good test. It has been now given to the company, and this will help for the human beings also. Now we come to other tests that it took several hours to do the PCRs. So we started using gold nanoparticle antibody based test for the detection of the spike antigen, and this is nothing but the gold nanoparticle, and on that is antibody coated, and as soon as this spike antigen of the virus tags to it, there is a change in the electric current, and you can detect it in a machine within a fraction of seconds. So this only shows, if you see the lower graph, that this is the COVID-19, and there are MERS virus or influenza virus or HIV, so there is no cross reactivity. So it is very specific. This is also being given to a company. Then similarly, what we have done is Japanese encephalitis virus jumps from pigs to human, and you need to check that in pig, and if you can detect correctly in pig and stop that there, then it will never jump to the human being. So we have developed a kit for that also, using I think graphene oxide particles, and it is also very specific. Now I will also come to another biotechnological importance for human concern, mastitis in animals. So milk, in several places milk is going, God knows how it is getting pasteurized, how it is packed, how it is given. And if the mastitis occurs in animal, several times it is an antimicrobial resistant Staph aureus, and it spreads to the human population. So technically speaking, to the animals also there is a huge loss. 7 crore livestock farmers and 30 crore cow or buffalo, and you can understand that 18 to 24 crore of them suffer from the mastitis. And if you see these colored bottles, these are nothing but the milk. These are normal milk, and these are subclinical mastitis, and as the mastitis grows, the color changes. So we cannot find anything in normal, but if we can catch subclinical mastitis, we can prevent them from going into a severe disease where the udder really looks bad and you cannot treat them. It's very, very difficult. Then the farmer keeps on whacking antibiotics, and you develop antimicrobial resistant bacteria. So to do that, it is better that instead of catching a clinical mastitis, which is visible by eye, it is better to catch a subclinical mastitis. And what we have done for that is just a simple iron nanoparticle based mastitis detection kit, where iron nanoparticles are made in a way so that they are going to catch hold of a small bacteria, or even smaller number of bacteria. And what you have to do is basically just put it and add a solution, and there is a clumping if there is a bacteria, and there is no clumping if there is no bacteria. And this is a field side test. You can go to a farmer's place and just take milk and do. So very small amount of antibiotics, you can treat the animal and prevent it from going into severe mastitis. It's a very wonderful thing, and it has now also been given to industry. So now, in addition to that, what we have done is we have established a DBT center for One Health at National Institute of Animal Biotechnology, and it encompasses 27 centers, 12 centers in the other states. I am calling northeastern states and other states, around 6 veterinary and 5 medical colleges, including AIIMS Delhi, AIIMS Jodhpur, and 7 centers in northeast states, 4 veterinary, 2 medical, wildlife nationwide, and 8 disease investigator units in northeast area, so that the transboundary diseases which are coming, they can be checked there. So we are training them, making them laboratories, giving them PCR machines, so that we can control that. So the aim of this consortium, I am the coordinator of the consortium of 27 institutions. It is to initiate and strengthen surveillance system of zoonotic diseases through existing and newer diagnostics, to have platforms from generating indigenous resources for developing detection kits, vaccine, animal models of disease, dig out pathways leading to AMR in Indian subcontinent, and tracking points for interventions to mitigate AMR and its aftermath. I would say it is the initiation where a One Health program with ultimate objective of establishing intersectoral collaborations This is most important thing. Among veterinary, medical, agriculture, environmental, wildlife, meteorological and other areas to detect, prevent and control zoonosis. We do not work with each other. Medical doctors do not work with veterinarians. Meteorological people do not have to do anything with the veterinary or medical science. But we all have come now on one platform. It was so difficult to catch hold of wildlife people. 3 institutes said yes and eventually rejected. With great difficulty, I could, at the end of first year, I could get a wildlife partner who can do the work for us, and it's very, very difficult. So in my view, this is a very big funding, and Dr. Katoj chaired it, and I am saying in my view, at present Dr. Y.K. Gupta monitors it as a chairperson. I told them that if I do not achieve 100%, achieve 35% of this objective, that is to bring this individual, make them friends with each other, make a common website where you report the disease which you caught urgently, and talk to each other about the symptoms. Several times doctor doesn't understand there's a brucellosis. But if he talks to a veterinarian, immediately he would come to know that these symptoms, maybe he's a butcher or butcher's brother working in any office, but he has got it from his brother. So we are doing it for the sake of generating a consortium in the India for the first time, where all these people will first become friends, know each other, have a common website, report the cases, and come together to make a good surveillance and stop the infections or outbreak wherever it happens. So bless us so that we go ahead with this. So this is the consortium in figure, how left side is the veterinarian centers and right side the doctors. So I will also say that there are plant biotechnologists sitting here, that One Health can never be addressed unless you take soil health and plant health into consideration. There's a lot of microbes are coming from there and foraging, and a lot of, as long as animals are there, I think human beings are there, they consume the plant parts. So it is very, very important. Now I will come to one of the real topics which will interest you. But I talked about how biotechnology is important for animal human. Now I will come to a topic which I may cover in 20 more minutes, I guess, is that it's very interesting for you, that there is an increasing need of therapeutic protein. Due to virus diseases or aging, due to various diseases or aging, some proteins are produced less than required in quantity in our body. So they are not produced well in our body. So we suffer from some disease or the other. And you understand, these human or animal proteins are today's prepared in vitro or in vivo and are used for these therapeutic purposes. That means you have to replace, give injections. Best example is insulin, or it can be if you have a Guillain-Barré syndrome, you have to give a lot of IgG to nullify the bad antibodies. And these proteins are needed to be replenished or supplemented to maintain normal physiological metabolic process in the body. Information about such therapeutic proteins are piling up. Now we know about them. Now we know this protein, if given, can be treated, a disease can be treated. And new avenues for treatments are also increasing. But what is the problem? Problem is that human or animal proteins are prepared in vitro or in vivo and are used for these therapeutic purposes, and it's very, very costly. Production of these proteins in large quantity with proper structure and biological activity is the new need of the future. So we know that if somebody is suffering from GB syndrome, obviously you have to whack in a lot of IgG, normal IgG, to prevent or recover the patient. If you have a severe osteoarthritis, you have to, now people are giving treatment of bone morphogenic protein 2 or 7, but these things are very costly. You don't see these being used in India, especially for arthritis. So let us say, what is the global therapeutic protein market? There is an increase in number of FDA approved biologicals. The one which is little dark is a small molecules, and one which is green is a large molecule. So what you will see that in spite of increase in market share, the production cost still remains very, very high. 71% money goes into the labor cost, and then equipment cost around 4%, facilities around 8%, and other materials, etc., 18%. So you have to have manpower, 3 shifts of duties, light, electricity, everything is required. Now, if you come to the reason is huge cost of infrastructure and establishment and manpower, etc. That's why these therapeutic proteins, although may be produced at cheaper cost, but sold at very, very high cost, which we cannot afford. So if you see in India, if you go to buy insulin, this is the just cost given, don't worry about insulin, but blood coagulation factor used in several diseases, each patient with a small cut required minimum of 2,000 unit if it's a hemophilia, and cost is around 20,000 to 40,000. Now interferon gamma-ferron is required for several treatment, and it is sold as rupees 14,000 per 80 microgram of one vial, and treatment depending on how it is, it can go up to 52,000 rupees. If you take the growth hormone, the general treatment is 7,500 to 15,000. And if you go to bone morphogenic protein, BMP2 is 2 lakhs per 1 milligram. So it's very, very difficult. So I really am surprised that how poor can afford in our country this kind of thing. So you will understand that India has very big population and life expectancy is 69 to 68 years, but 269 million is we have 21.9% of the total population. So what is more important for me is that I think the things change in this slide, that we are using only 1.6% of the world's market and 3.2% of the total pharmaceutical market. Ideally, looking at the population that we have 1/6 of the world population, India should be holding 15 to 20% market. We do not. It is not that we do not have diseases. It is that we don't have money. People can't afford it. So there is a requirement of various therapeutic protein in huge amount, but there is a need to make them available at cheaper cost. So what is important that when you want to generate this therapeutic protein, there are various ways: recombinant bacterial expression system, plant-based system, animal culture-based system, or yeast-based system, and a lot of them are being used in the industries at present. Something is called as animal bioreactor. That means if you can generate these proteins in the blood, urine, seminal plasma or milk, people have attempted, you can make them cheaper. So these can be done by generating a transgenic animal where you have inserted the gene with proper promoter, and if it is a targeted to whichever samples I have talked about, this can be expressed in milk, seminal plasma, urine or blood, and then you can purify. Now why? Because the conventional method of production of therapeutics, you need a huge industry, infrastructure, electricity, land, labor, labors in shifts, license, GMP facility, etc. But if you go to an animal, it's an animal bioreactor standing there, and this cow, buffalo or goat is everything, your light, electricity, infrastructure, whatever you call. So in milk, it can be produced at much, much cheaper cost. So since Western researchers have isolated cow and goat milk protein promoters to drive therapeutic proteins in milk, in the era of WTO and TRIPS agreement, India may need to have its own source. So hence buffalo genome was used by us for isolation of the milk specific promoter. So we will be a little different and we can go ahead, otherwise sometime we work and somebody say patent is there, so you can't work. So we isolated buffalo beta casein protein. I will not go into detail, but it is known that buffalo beta casein protein ideally should be very active. So we have isolated buffalo beta casein protein gene, and that is a promoter, and along with the exon 1 and 2. And then we tagged, I am not going into details of proving that we have isolated, but then we have tagged EGFP. First we wanted to check that with a buffalo beta casein promoter, it should drive any protein tagged behind it only in the udder gland, nowhere else. So that was the most important. So we tagged EGFP first, and then after tagging EGFP, we have gone and we have tried to use this mammary gland specific promoter in a cultured mammary epithelial cells of buffalo. And it was expressing EGFP. So we knew that it is working at least in buffalo mammary epithelial cells. Then we generated a transgenic animal. So we discovered a technique in 2008, which is a very good technique, that rather than doing traditional embryo and all those, which is very cumbersome and costly, we just insert genes into the testis and electroporate the testis, and we generate, just put it for mating and generate transgenic animals. So we generated transgenic animals. So this is the udder gland. You can see how transgenic animals' udder gland is fluorescing green, how normal mice's gland is not fluorescing, because this is not a transgenic mice. So after we have done that, we knew that this is driving, and our job was to go to the large animals to do this job. So I will just show you that EGFP is shown, is a larger image only in the udder gland, and when you kill the animal and to do the western blot of these glands, you find that 3 animals, which are the transgenic animals, expressed EGFP in their gland, and the wild type animals did not. Then we thought, is there a leaky expression in other organs? So we killed this animal. We have taken liver, breast, kidney, spleen, heart, and brain, and we found it is not expressed anywhere, but it is expressed only in the breast, that is the udder gland, proving there is no leaky expression of this promoter, and it is not driving the gene in any other place other than the udder gland. So the difficulties with the transgenic animals are there. One is cost, very costly. We want to make large animal for more milk, goat, buffalo, cow. Making transgenic mice is very easy. But having superovulation and hundreds of eggs in a cow or buffalo is very, very costly affair. So the cost is written here, the red circles, we need not read it. And it is also time consuming. And most importantly, the germ cell transmission, GMO. So we are making a transgenic animal. So we are seeing what is happening to mustard and BT brinjal and all. So it's difficult. I do not know whether I make it and try to produce therapeutic protein. Somebody will say, no, this is GMO. I will not allow this to be propagated further. So I will have this problem. So we started thinking, our whole group started discussing. So transgenesis is germline integration. So what we thought that non-germline integration, how can we do the non-germline integration? So a beautiful thought came in mind, which when we went into literature, we found some people have given that thought also. Instead of generating a whole transgenic animal, you go and inject the mammary gland and whack in your gene, but you have to know the mammary gland development very nicely, because unless the cell is profusely dividing, your gene can never be integrated. So that's very difficult thing, but we started working, and we did virosome mediated delivery of transgene in the mammary gland. So for doing that, basically from the virus, Sendai virus, we remove the nuclear material and actually insert the other nucleus or nuclear material into this Sendai viral, removing original and inserting other. So how that is done, I will show. So we do the Sendai viral culture in the egg, and then we take this and actually treat it so as to really rupture, or the detergent solubilization will make the nuclear material and the lipids separate, and then we centrifuge, remove the nuclear material, and then what we do is insert the DNA of our choice in this solution where original nucleic acid is removed, and this is the human DNA containing nucleic acids. And then when we add it and treat it with your detergent removal with SM2 biobeads, what we find that they again aggregate, the virosomes aggregate, but in between now tagged is our gene of interest. And if you now use these virosomes for transfecting the other glands, you will be able to really but for that you need to know the other gland development, which we studied very well, and we did several experiments. But finally we have come that in mice it is the 19, T21 is the parturition, that if we can insert the gene in the udder gland, we can successfully transmit. Before that, so many people may have a question in their mind that if you are making this into udder gland, it is possible that if you make huge amount of human gamma interferon, it can go to the blood and can circulate in the body of the animals. So we just want to clarify that actually what happens, that blood is outside. Blood doesn't go into the milk gland, and blood comes from the top side and flows like this. And whatever is produced in the milk gland, it is an exogenous gland, it exits. It doesn't go. Otherwise, mother will have a lot of lactalbumin in her blood during when she is feeding. It does not happen. What we will do, we will use this area to really transfect the gene of our interest, and we do that. And then let us see that EGAP with promoter is there. Luckily, if this works for us, how will we do? We have done it. So this is the teat, you can see the teat, and in this teat we will insert a needle. Can you see? Very finely it is inserted, and we can directly transfect or inject at this moment the promoter driven with EGFP, for example, and these are the teats. And if you take a cross section, these are the virosomes sitting inside the each duct, milk duct. And if you are lucky, what will happen, that these virosomes will transfect the cells, which it does, and the DNA will be transferred. And if the cell is dividing, this DNA will integrate and cell should turn green. It does turn green. And in fact, in vivo, we find that the udder gland turns green. So remember, first I have shown that was transgenic animal. This is not a transgenic animal. This is a only udder gland manipulation. And we find that as compared to the wild type black, we have the two transgenic glands, and which are showing green line. Now we have done EGFP expression in transgenic positive female, which we had. And now directly transfected animals, 3 of them, we found similar level of expression of EGFPs there in them, proving that this may work for us. This is the actual cellular staining, and it does work. And then we have again done the same thing. Kill the animal, taken heart, breast, liver, kidney, spleen, brain, and abdominal skill. Now somebody can say while you inject, it can go in several places. It does not. It is only specific to the udder gland. Now, you will remember that till then I was talking about EGFP. So instead of that, we need therapeutic protein. We are not working for EGFP. So we tagged now human gamma interferon in pink color, you can see in our construct, and generated such animals, and we found that in non-injected gland there is nothing, but in injected gland you can find expression of human gamma interferon, and this is the western blot expression as compared to wild, and this is the expression when we did an ELISA using the minced supernatant from the breast gland, and ultimately we have taken the milk. There are various ways of finding the milk secretion, and we found that milk is also expressing human gamma interferon as compared to the wild animal. So this was really a success story. We published this, and in addition to this, we have also done the work with the rabbit, because people say, you'll be surprised to know, more than cow or goat, rabbit is the best model for protein production. They are neat and clean, they repeatedly produce babies very fast, and they can give you more milk versus the feed they are taking as compared to cow. So if you want really to make a big industry of therapeutic protein, rabbit is the best one to do. But anyway, that's the reason we went into rabbit, and we could generate doing the same thing in rabbit, human gamma interferon in large quantity. Now this is human gamma interferon. Similarly, we need FSH for superovulation of our cattle. 35% of the buffalo are infertile. And several times nowadays we have got avenues using SNP chips to isolate good cows and good buffaloes in the beginning. You would like to superovulate them and make more babies out of them, and then sell them or distribute them and make a big herd of the good generations, and do not use the bad ones. That you can know by SNP chip. I will come to that later. But for that, what you need is lot of FSH injections for superovulation, for getting more eggs. Sometimes just FSH injection make them fertile also. But cost of FSH, you can see, for single treatment of an animal, it will cost around 27,702 Indian rupees. So a farmer can never afford this. A farmer, if you say you have to take a vaccination for 50 rupees, and you have to go 4 villages away, he doesn't go. He says, Mera dehadi mara jayega. I will not earn today if I go. The cow cannot go on a flight. Na? So he has to walk and come back. So farmer's situation is very bad. He cannot spend 50 rupees. So we wanted to do this. So what we have done, I will in nutshell show that we have generated very, very difficult thing, because in FSH is not a single protein. Alpha and beta has to dimerize, and then they have to become biologically active. It's a mess. Several of us tried, and very, very difficult. But with the grace of God, I will just give you a glimpse that we could generate FSH transfected cells supernated in CHO cells. We could generate that in a single cell. You can create full FSH. Now whether this FSH is and we have done the ELISA also, and then whether it is bioactive or not, that was very, very important. So since we do a lot of testicular cell cultures, this is an intact testicular culture, fraction of testes, and when we give ovine FSH 50 nanogram, we see change in stem cell factor, supposed to be, because it is biologically active. And when we give our recombinant bovine FSH, what we find that stem cell factor is again increased, indicating that whatever FSH we have made is biologically active FSH. And remember, we just use ours 5 nanogram as compared to the standard which is purchased ovine FSH 50 nanogram. So it's comparable, more than that, and good. And same work we have generated in rabbit, and in rabbit milk also we are finding very good 1600 milli-IU per ml of the rabbit milk, not bad. This is very, very good news for us, and we are very happy that we have generated at least one human and at least one usable animal protein for at a much, much cheaper cost. Now the question comes, here I will say that biggest thing is the cost of virosome. So eggs, every virosome culture doesn't yield to if people who work will know, they have to throw all 200 eggs and again do the culture. It's costly. Then you have to get, the yield is very less. Again farmer will come ki sir, मैं 200-300 रुपए भी नहीं दे पाऊंगा. So we were thinking about that how to transfect an udder gland at a cheaper way, much cheaper way. So we went into chemical way, and we have what we have done, that we have added hexanoic acid to the branch polyethyl imine, which is usually used for transfection of the cells, and this is called as the hexanolated branched polyethyl amine, and we call it FS6 BPI. And when we did this and transfected the udder gland, what we found that at a NP ratio, so what is NP ratio? Is ratio of the positively chargeable polymer amine groups to negatively charged nucleic acid phosphate groups. That's how you know how much is tagged. And at a ratio of N is to P11, we got a wonderful result. So you can see so many green cells, they are all transfected. And mind well, now my lab and NIAB whole institute doesn't use lipofactamine 3000 or something. They use this, which is extremely supreme. And some of the cells we were failing to transfect, like Sertoli cell never gets transfected. We can transfect with this compound. So this we have patented, applied for the patent, I would not say patented. And we found that this not only gives the green fluorescence protein expression, but now in HEK 293 cells, MCF7 and goat mammary epithelial cells, we can successfully transfect them, and they are producing human gamma interferon. This is a graph of human gamma interferon. So it works for us at a much cheaper rate, and we will use it for transfection of the udder gland, and that's what we did with the goat. And we found that if you use this for transfecting goat udder gland in goat milk, you can see these are the amount of the human gamma interferon, but it comes down by 72 to 96 hours, because this must be episomal expression. So we have to wait for a period of a month or two, and then again when she gets pregnant and delivers, we have to see whether it is coming in the milk or not. So this is what is our plan, that if we can do this job, now we'll go to large animal, that is goat, and try to do that. You can human or animal genes, if we can insert in the cow and udder gland, and maybe 14,000 vial can be one day produced at a cost of 200 rupees a vial. This is our dream. I do not know whether that will come true or not, but there is another important catch. If you generate a transgenic animal of such kind, suppose the udder gland doesn't work, in case of mice, you put for mating, 12 babies are born, cut the tail, do the DNA, and you find out of 12, 3 are transgene positive. In case of buffalo, 10.5 months you have to wait. One baby will be born. God knows whether it is transgenic or not. So it is extremely important that if we do testicular transgenesis, if the GMO is allowed in large animals, we need to select the transgenic sperm and then do IVF and transfer, or do the artificial insemination. So there is a guarantee that after 10.5 months, a transgenic baby will be born. So what we have done, in the membrane surface, there is something called as basicine, which hangs out. So we have tagged GFP by genetic engineering to basicine and generated a sperm where ideally green thing will hang out from the surface, and you can fax sort it or magnetic bead sorting it, and then you can use it for artificial insemination with a guarantee that there will be a transgenic pup. So this work is parallelly being done now, thinking that this may be a problem of the future. And you can see on the left hand side this green sperm we made, but before that basicine is like this, and we have tagged the EGFP so that basicine should look like this, and we have generated transgenic animals where basicine should ideally hang from outside, and it is, I am saying you, these are the photographs of those. So, and what will be done basically, I will go very short, that this testicular transgenesis, if we produce babies, and we have to sort the sperm, and here some of the sperms are showing green because they are transgenic. So it will be dual transgenic cassette, one transgenic cassette for expression of basicine on the top of the sperm tail, tagged to the original transgenic cassette, buffalo beta-casein promoter with human gamma interferon. So it's one. If there is a GFP positive sperm, we know it's carrying my gene of interest, that is human gamma interferon also. And we did that. And our job is to really fax sort it, and green sperms can be taken, or do the magnetic sorting, that also you can do. And then green sperm will be taken, and that you can use either for artificial insemination or IVF or whatever you want to do. And then you can have guaranteed transgenic animal with this. So this is just a figure showing that these are the various things where it is showing that only membrane, this is the actual expression of the EGFP, so the second antibody is red, that only in the membrane this is shown, and this doesn't go anywhere else. Lastly, I will show that we have also made something was pending from long time, that is to generate the Indian cow's genomic chip. So when I was saying that using the chip you can identify good or bad animal in the childhood, from there you can take testes or sperm or gene or whatever, or elite bull for artificial insemination, or elite the cow for taking superovulation. For that now NIAB has generated a chip. We called it, this is the first SNP chip of the country, taking 43 breeds of the cow as a dictionary. It has got 8 lakh SNPs, that's where it is biggest of the world, and this is a proud for us. And we can now choose best herds out of this. We can choose the heat resistant or disease resistant animals also out of this. And most importantly, we can choose out of 100 bulls in the childhood, you can select these 3 are the best. So do not pay attention to others, grow them, use their semen for artificial insemination. That all can be done, and with NDDB we are doing this work. And Minister of Science and Technology on 13 August has really released this, and we are proud of that. So I will come that this all is about One Health, that we must work towards One Health. So see how we animal biotechnologists are trying to generate therapeutic proteins for human using our technologies, or good kits which can be used in human and animals both, or a One Health center where we are thinking that doctors, medical, veterinary, wildlife people should come together and work. And the beauty is that we think that they gave us and they think that we are the culprit, because it's a zoonosis and reverse zoonosis, both are there. Human also give them this kind of diseases. So it's very, very difficult time. So though the problem is big and ground is vast, human beings need to collate all their knowledge, including biology, machinery, artificial intelligence, for detection, prevention, and curtailing health effects of the zoonotic and reverse zoonotic diseases, inclusive of equal importance to human and animal health. And I would say we will work for each other, animal health for human welfare, that should be our motto, and biotechnological interventions for conserving each other should be our ultimate goal. Thank you.
Yeah,
Ah.
please.
Please.
Yeah, go ahead.
I can.
Go ahead. You can use the mic.
So your One Health initiative is a great initiative, I think from NIAB what you have taken, that's wonderful. But particularly zoonotic disease, vector biology also play important role. Particularly in the time of global climate crisis, when you see that vector life cycle changing very fast, that's probably one of the reasons we see so many emerging re-emerging pathogens. But how that fits here?
Actually, each center is expertise in vector biology, disease dissemination, as well as detection. Because these are some ICMR centers, including Gorakhpur, including say Dibrugarh, and all veterinarians really work with the vectors also, because they have to look after their own animals, and vector is in between human and animal. So yes, they have expertise of that also. So it's inclusive of all. But let me say, the whole One Health is divided, in my view, into three different categories. One is epidemiology and forecasting. So the environmental people should be in meteorological. They should not only detect, they should also be able to forecast that if this has happened in, for example, in Jalpaiguri, so the way this season is going to be, very soon it is going to come to Raniganj. And immediately they should cordon off or take the disease. One is epidemiology and forecasting. Second is preparedness. That means if any disease comes, you must have all sorts of platforms like vaccines, etc. done. And third is therapeutics. So I think people, what we are working on, epidemiology and forecasting, disease detection, some kits, etc. Others should work on other. This is a very huge subject. One set of group cannot work on all. That will be too much. They won't work. Gopal.
Yeah, Dr. Subeer, I think it's excellent talk and a lot of biotechnology product-based research. So I have specific question. Interferon gamma alpha IFN and follicle stimulating hormone FSH as well as BMPs, what you talked about. There is a difference in sequence identity among amino acids between human and animal.
Yeah.
And when you are expressing in that animal system in a transgenic way, in terms of their activity when you are planning to apply as a biotherapeutics in patients, in terms of microcontamination, in terms of their biological activity, and in terms of their sequence identity, how close they are
So
towards that recombinant, this class of protein which has been manufactured by multiple biopharmaceutical company in India.
So what we are making is nothing, but when we make human gamma interferon, it is human gamma interferon gene. And we are just expressing human gamma interferon, and we have checked, we have taken the cells from Mohanwani, and in human bone growing cells, stem cells, we have seen that it does the what you call calcification of the cells, alizarin staining, and it works. That is for
you are talking osteoblast.
Bone morphogenic proteins. I am sorry.
Osteoblast cell
Yeah,
line.
osteoblast for bone morphogenic. Likewise, we are doing bioactivity in the species specific bioactivity. with the cell lines.
So is it comparable that whatever these recombinant protein available in the market by other parties and with along with your this protein?
No,
what
that
you
we have not
manufactured?
compared. That we have not really compared in terms of cost, but I am pretty sure it will be much cheaper than them.
I'm
I have
talking in-
compared one like ovine FSH. You
I'm talking
require.
in terms of activity.
Oh, I am
in
saying
terms-
you. So, like for cell cultures of the rodents, they use ovine FSH. So, for 50 nanogram ovine FSH, we are requiring only 5 nanogram of bovine FSH. So, there the cost comes down. And secondly, ovine FSH is very costly as such, and this is made in house.
So excellent talk and wonderful work. Compliments. So wanted
Thanks.
to know if your institute is working on the vaccines for the Asiatic lions and distemper. I'm from the Wildlife Institute of India.
Yeah,
So
yeah. So I know
Any
that
progress
this,
on
this is not the work of NIAB, but I think IVRI has taken up this work. I personally feel, but since I have gone closer to that area, probably my institute can take up. Because the where I have joined now recently, Gujarat Biotechnological University, there they have got the medical biotechnology and animal biotechnology both, along with industrial and environmental, etc. But that's a interesting thing we can take up. But vaccine thing, I think IVRI is working. Indian Veterinary, Izzat Nagar, not us. Yes, sir.
Excellent talk. I have one curiosity. The virosome-based udder cell transfection, there is no selection marker used, right?
Sir, no, we first used EGFP
Yeah.
to check it is udder gland specific,
Right.
not going to brain, liver, lungs. And then once you confirm that, you tag anything behind it.
No,
Well,
that's what I understood.
No,
My
we
que-
have not. No,
My
no.
question was then what
No.
percent of cells get transfected?
What is the?
What is the percentage of cells getting transfected?
In the udder gland?
Yeah,
I think it's, you have seen it's whole green.
that's the reason I asked. that.
Yeah,
Is
we
it
have
whole-
not really calculated that because we are more interested. We are more interested कि दूध में कितना आ रहा है.
Yeah, that's true. That's true.
So that is what is our
Okay.
interest. Because the let us say goat and cow, beta lactoglobulin promoter and beta casein promoter, they have isolated. So हमको तो वो काम करने देंगे नहीं. Buffalo क्योंकि हमारा indigenous है. So what we have done from the genome, we have taken the promoter with exon 1 and 2, cut and added EGFP behind it. So since it is a beta casein promoter, beta casein doesn't express in any part of the body other than the udder gland. So even if you make the whole transgenic animal, beta casein can only get expressed in the udder gland. So that's how it is specific, gland specific. But that we also checked. Sometimes क्या होता है कि हमने beta casein को देखा यहाँ है, वो glial derived neurotropic factor GDNF, वो तो कहाँ दिखाई दे रहा है शरीर के several places. Like that we checked several organs कि is it जो आपका question है कहीं और भी तो नहीं express हो रहा है. So what we found major organs it does not express. Because otherwise it will be havoc. अगर brain में gamma interferon secrete होने लगा तो problem होगा. Thank you, sir.
There are no questions. Let
Thank
me
you, sir.
again congratulate Subeer for his excellent talk.
Thank you, thank you. Thank you, sir.
Yes. Thank you so much, sir, for chairing this session. And indeed, it's our pleasure to have you here. Yeah, an eminent scientist, a fellow of 4 academies, yes, J.S. Bose Fellow, and visiting professor at several institutes abroad. Thank you so much, sir. So now we have come to the end of session 4, and now we'll break for the lunch. And after lunch, we are going to have a lecture. Professor Vishwanath Memorial Lecture at the YVS Murthy Auditorium itself, and followed by the tea. And by 3:45, we will be again here for the next lecture. Thank you so much. We will now break for the lunch. Please join us in the YVS auditorium outside the premises of the YVS auditorium. Thank you so much.